Hello all, I'm quickly going to talk through the exam questions that you did last week. I'm not going to spend a lot of time because I'm going to put the mark scheme on the drive so you can just mark it yourself. But I just wanted to go through the calculation more than anything else. Some people were asking about this when you were doing it. It says, describe how farming cattle as a source of milk could have led to an increase in lactase persistence. So when we're looking for a good answer, you need to say something about a mutation causing the new allele. You need to describe, you need to give a reason why this persistence is a good thing relating to a nutrient. So you need to name a nutrient that is going to be beneficial to you for drinking milk. So that could be the glucose, the galactose, could be some protein. And then coming straight into our sort of natural selection type answers, individuals with the persistence are more likely to survive and reproduce. So it's really important that you are combining these two ideas, survive and reproduce. You wouldn't have said anything about directional selection because we've only learned about it this week, but it's a 4-mark question. But what you really should be saying, and this is one of the most important parts to remember, is that the frequency of that allele is going to increase in the population, and because it's being passed down to the offspring and then the offspring in the next generation. Okay, don't say number of alleles, it's the frequency of the allele in the population. Okay, use the information provided to explain why the number of people showing LP would rapidly decrease once selection for this condition has been established. Well, firstly, it's a dominant allele, so if somebody has it, it's always expressed. Okay, and that's the reason why. And so that's using the information. So they've given you information, use the information provided. The information that they've provided is the fact that it is dominant. If it's dominant, it's going to rapidly increase. If it was a recessive allele, it wouldn't increase that rapidly. You wouldn't see it because you'd need to have 2 copies of the recessive allele in order to actually see the phenotype. Lactase persistence is caused by mutation in DNA. So I said to you, this one is tapping into year 13 work. The idea is that the mutation is actually in the promoter region for the transcription factor. And so that the lactase gene continues to be transcribed. But don't worry if you didn't get that one, because that is very much tapping into year 13 work. Question 2. This was a description of how you make a dilution. So when you're describing it, you're using words. So add 1 part of the culture to 9 parts of liquid to make a 1 in 10 dilution. Mix it well or stir. So can you see they're very particular about the actual method? So when you are going to do a dilution, you mix them, then you stir. Okay, so be aware of this. Be aware of the level of detail. And some of it seems really simple, but you leave it out, obviously you're losing marks. And then repeat using 9 parts of liquid and 1 part of the 10 to 1 dilution, and then do the same thing to make the 1000th dilution. Okay. Otherwise, you could just short circuit it and use 1 part of the 10th dilution to 99 parts to make up the 1000th. Either of those is absolutely fine. But out of a 3-mark question, you can see you're getting a mark for saying mix it. So keep these things in mind. Paper 3, practical. They want to know that you can do these practicals. Using an optical microscope, the student determined that there were 15 cells in 0.004 millimeters cubed of the 1 in 1,000 dilution of the culture. Calculate the number of cells in 1 centimeter cubed of undiluted liquid culture. Some of you are going to hate these sorts of math questions. Some of you are going to love them, but I wager most of you won't be a massive fan. But you need to think about what's being asked here. So I'm going to try to change my screen so that I can share the answer with you. Okay, so what are you given? You're given that there are 15 cells in 0.004 millimeters cubed. And you're also told that it's 1 in 1,000 dilution. So the questions you're asking yourself, 3 questions to get this calculation done. How many cells are there in 1 millimeter cubed? How many cells are there in 1 centimeter cubed? And then how many cells are there in 1 centimeter cubed of undiluted culture? So the first thing we do is acknowledge here that, lost my little spotlighty thing. Still not there, where is it? Come on, okay. So that we have 15 in this very small volume. This very small volume is 250 times smaller. 1 divided by that number is 250 times smaller than 1 millimeter cubed. So if there are 15 in this volume is 250 times smaller than 1 millimeter cubed. Then to find out how many in 1 millimeter cubed, multiply 250 by 15. And that tells me that I've got 3,750 in 1 millimeter cubed. So now I want to work out how many there are in 1 centimeter cubed. So hopefully from maths, you remember that there are 10 millimeters in 1 centimeter. But we're not looking at millimeters and centimeters here. We're looking at volume measurements. So we're looking at millimeter cubed and centimeter cubed. So, but 10 millimeter cubed in 1 centimeter cubed. So actually it's 1,000, because 10 to the power of 3 is 1,000. There are 1,000 millimeter cubed in 1 centimeter cube. So if we have got 3,750 cells in 1 millimeter cubed, we've got 1,000 more in a centimeter cube. So in 1 centimeter cubed, we've got 3,750 multiplied by 1,000. So that is 3.75 times by 10 to the power of 6. Please remember to be writing your large numbers in standard form. The last thing we've got to tackle is how many are there in the undiluted. Well, we know it's diluted 1,000 times, so we simply multiply it by 1,000. So in undiluted, we have that many. So if we multiply by 1,000, we have 3.75 times by 10 to the power of 6 times by 10 to the power of 3. We remember from maths, when we multiply these together, we add the 6 and the 3. So the answer is, 3.75 times by 10 to the power of 9 cells. So hopefully that helps you with the maths there. Let's see, back to the question. Okay, this one here. Explanation for the student's decision. What did he do? He decided not to use this dilution. And the reason he decided not to was because the count was unlikely to be accurate or repeatable or reproducible or reliable, because there are too many cells, or because cells were overlapping or not spread out. Okay, so you need to say what, you don't just say it is accurate, it is reproducible, it isn't reliable. You need to say what is the count. So counting them is, because there's too many cells to count, basically. Okay, suggests one reason why bacteria-resistant tetracycline are more common than bacteria-resistant streptomycin. So that's 2 marks. So you've got 3 options of answers that you can get for 2 marks. So you could say that tetracycline is used more often in higher doses, so that resistant bacteria are more likely to survive and reproduce and pass on their allele for tetracycline resistance. Okay, so it's suggest and explain. Suggest, explain. Okay, here's the other one. More higher frequency of mutations, so genes are passed on to more bacteria. Suggest, this is the suggestion, there are more mutations. This is the explanation, so the gene is passed on to more bacteria. Or tetracycline is used over a long period of time, so there is more time for a chance mutation to occur. Suggest, okay, this is your suggestion, and this is your explanation. So any of those 3, but you have to give a suggestion and an explanation. In recent years, these farms have not been given, farmhouse have not been given tetracycline in their field. Despite this, the percentage of bacteria-resistant tetracycline has remained constant. Why? Well, because there's no selection against the resistant bacteria, or the that bacteria pass on the resistance gene when they reproduce, and so on and so forth. So you can read through that. Okay, and you'll find that in the mark scheme that's on the system for you to look through, it's different to this. There are examiner's notes present, so you can access those and read through what kind of mistakes students commonly make. Okay, I hope that helps.